Hey everyone, welcome back to Window Seat Studies. I hope you all are doing well. So, we were discussing about cancer biology, in which we have already discussed about the differences between malignant tumor and benign tumor. We also discussed about properties of cancer cells. We also learned about the hallmarks of cancer. So, the last lecture was kind of an introduction lecture where we covered all of the hallmarks of cancer in brief. And from today onwards, we are going to cover all of those hallmarks one by one, step by step in detail. So, we are going to learn about the mechanisms by which or the means by which those tumor cells they are able to exhibit those certain traits. So, we are going to cover two hallmarks in this lecture, which are very important, or let's say they are the central hallmarks of the cancer. So, we are going to discuss about how the cancer cell they are able to sustain a proliferative signal as well as how they are able to evade the growth suppression. So we know that cancer cells show the uncontrollable proliferation. So the very first hallmark that we are going to discuss in today's lecture is that how do those cancer cells or the tumor cells they show sustained proliferative signal. So, as of now, we already know that the tumor cells, on the contrary of normal cells, they need a continuous signal so that they are able to divide. Okay, so what happens is that in normal cells, there is a very tight regulation where there is a very tight control on the production and exhibition of the growth promoting signals, so that a normal cell undergoes division until the Hayflick limit and then it undergoes the programmed cell death. But this doesn't happen in the cancer cells. The cancer cells, they are uncontrollably growing cells, so they need a continuous supply of the growth promoting signal. And so now we are going to learn about the widespread mechanism, the very sure mechanism by which this continuous signal in the cancer cell is maintained. So there is a group of genes which we call as proto-oncogenes, okay, which are central to this hallmark. So the mutation in proto-oncogenes, which turns them to the oncogenes, okay. So upon the mutation, the proto-oncogenes get converted or get transformed into the oncogenes, and those oncogenes they are going to lead to the cancer. Okay, so the mutational alteration in proto-oncogenes converts them into the active drivers of the cancer, which we call as the oncogenes. Okay, so those group of proto-oncogenes, they are the genes which are they play major role in growth promotion. So let's say let's assume that let's call them as the growth promoting genes. Okay. So so proto-oncogenes they encode for the proteins which are going to either rely the growth signal or they are itself the growth factors. So proto-oncogenes they produce growth factors. Okay, so those are the group of genes that encode for growth factors, or they could encode for growth factor receptors, or the transcription factors which are important in cell division and cell differentiation, or they can also code for the signal transducers. The signal transducer which are important for the growth or for the cell proliferation or the cell differentiation. So the proto-oncogenes, I hope it's clear that the proto-oncogenes is the group of genes which is going to rely the growth signal. Okay, so those are the genes they can code for the growth factors, they can code for the growth factor receptors, or they can code for the transcription factor which are important for cell proliferation or cell differentiation, or they can also code for the signal transducer. So in short, they are the genes which are going to which are important for the growth promotion. Okay, so now there's a controlled expression of those growth factors. There's a controlled expression of the proteins encoded by proto-oncogenes. But now when the mutation occurs in these group of genes, they get converted into the oncogenes, and this mutation is always the gain of function mutation, because now so you need a continuous growth signal, right? So for the uncontrollable growth you need a continuous signal, and so those are growth promoting genes. So this signal there should be the overexpression of genes. Okay, so that is why the gain of function mutation is important, because okay, I hope you know about gain of function mutation and loss of function mutation. So it has very literal meaning. So whenever a gene undergoes a mutation which results into the loss of its function, it is known as loss of function mutation. On the contrary, when a gene undergoes certain mutations such as point mutation, deletion, or insertion, which leads to the increase elevation of the production of whatever proteins that gene is encoding, that is what we call as gain of function mutation. So there are, let's list down the causes of gain of function mutation. Okay, so let's list down what kind of mutation proto-oncogenes can undergo so that they can lead to the cancer. So the very first thing is that they can undergo the point mutation, or they can undergo insertion or deletion. Okay, so any of those mutation which is going to result into hyperactive gene product. Okay, so this is very first cause of the gain of function mutation. So on the contrary of the normal gene product, which is functioning normally, when you have a hyperactive gene product, the function, whatever function that gene is going to produce, that will be elevated. So that is what we call gain of function mutation. So whenever there will be hyperactive gene product, hyperactive gene product is produced by those proto-oncogenes, it is going to result into the relay of the extra there will be elevated growth signal. So now that is going to lead to the uncontrollable growth. Or there could be certain mutation in the promoter region. So you know that the promoter region is very important for the transcription of any gene. So if there is a mutation in promoter region which is going to result into the elevated transcription, that can also lead to cancer. So let's say that the promoter region that is mutated such that it results into elevated transcription, this is also going to result into the uh, emergence of cancer. Okay, other kind of cause for the cancer could be, there could be the extrachromosomal copy. Okay, extrachromosomal copy of the proto-oncogene. So let's say that normally there's only one copy of the gene, and one copy of the proto-oncogene. So this is proto-oncogene, okay, and in the normal condition there's only one copy, but due to certain mutations there is an extra chromosomal copy besides that. So now when you have an extra copy of that gene, obviously the production of the proteins that is going to result into the growth, that will also be elevated, and that is going to result into the gain of function mutation. So that is going to result into the gain of function of the proto-oncogene. The other kind of mutation could be, or the let's say there's I hope you know about the chromosomal translocations. So chromosomal translocations, they also result into the gain of function. So let me explain it in a bit how that can result. So let's say this is a chromosome on which there's a region here, and this is an hyperactive region. Okay. So the region which is usually hyperactive, which results into the production of proteins, overexpression of proteins. And while here, this is the location of our proto-oncogene. Okay. Okay, so let's say this is the location of our proto-oncogene. Let's say that there's the other chromosome, and here is the location of our proto-oncogene. Okay, so this is where the proto-oncogene is related. So now there's chromosomal translocation, and this proto-oncogene translocates into the hyperactive region of the other chromosome. So when this proto-oncogene will translocate to this hyperactive region, now as it is here at, so now it is going to result into the overexpression of the whatever proteins that proto-oncogene is going to produce, and this will result into the gain of function. So these are the different these are the different reasons by which the proto-oncogenes they can exhibit the gain of function mutation, which is going to lead to the cancer. So I hope with this it is clear to you all how those mutations will lead to the cancer. So generally there are dominant mutations in the proto-oncogenes which will convert them into oncogenes, which are the important cause of the cancer. So with this we have covered the very first hallmark of cancer. So let's cover the second hallmark of cancer. Like let's learn about how those cells they are able to evade growth suppression. Okay, so we have already ensured that there will be, so okay, in normal cell there will be the growth promoting signals as well as some growth inhibiting signals. Okay, and the tight regulation of those two kind of signal, the positive signals and the negative signal, that is going to result into the cells dividing to from 40 to 60 times. So they will be dividing till Hayflick limit and then undergoing the cell apoptosis. But the cancer cell, they are the cells which are going to show exhibit the uncontrollable cell division, right? So they ensure two things. First, they will increase the growth relaying signals. So we have already discussed about how they sustain the proliferative signals. And the next job that they have to do is that they need to inhibit those inhibitory signals. So they are going to make sure that there will be inhibition of the growth inhibiting signal. So they will ensure that there will be no relay of growth inhibiting signals. Okay, so and that is how so that is how they are able to evade the growth suppression. So the widespread mechanism for evading growth suppression is mutating the tumor suppressor genes. So there is a group of genes which we call as tumor suppressor genes. Okay, so those tumor suppressor genes, they have antagonistic roles to the proto-oncogenes. So they are exactly having the roles which are opposite to the what proto-oncogenes does. So proto-oncogenes, they are the genes which are important for growth proliferation and for cell proliferation and cell differentiation. While the tumor suppressor genes, as the name suggests, they are important for suppression of tumors. So those are the genes which are going to inhibit or they which are going to limit the cell growth and cell proliferation. So those are the genes which limit cell growth and cell proliferation. Okay, so they both of those genes are antagonistic to each other. So in normal scenario, when the tumor suppressor genes are working fine, it is they are going to inhibit the cell proliferation, and that is how they will ensure that there is no tumor formation. But when these tumor suppressor genes they undergo loss of function mutation, so let's say that there is some mutation which results into them losing their function, which we call as loss of function mutation. That is going to result into no growth inhibiting signals, right? And when there is no growth inhibiting signals, now the it's like the tumor cell have the free pass to uncontrollably grow. So loss of function mutation in the tumor suppressor gene that will lead to the cancer. Okay, so this is exactly opposite to what we learned in the proto-oncogenes. The proto-oncogenes, upon gain of function mutation, they will turn into the oncogenes, which are usually the dominant mutations, and they are going to result into the cancer because they are oncogenes are the active drivers of cancer. While on the contrary, when the tumor suppressor genes they show the loss of function mutation, they are going to result into the cancer. So let's sum so yeah, let's so there are two types of tumor suppressor genes. Okay, the first one we call as gatekeepers, and the other ones are known as caretakers. Okay, so let me explain what two types of tumor suppressor genes are. So those the tumor suppressor genes, they can they can suppress the growth by two mechanisms. Either they can directly involve in the cell death, or they can indirectly promote the genomic stability. So what happens is that they can directly, when they directly in they're directly involved in cell death, the programmed cell death, which we call as apoptosis. So now, as the cell has died, so it is no longer proliferating. So this ensures the tumor suppression, right? So gatekeepers are the tumor suppressing genes, means they those genes they are directly involved in the tumor suppression. So they are directly involved in tumor suppression. Okay, while the caretaker genes they do not directly suppress the growth, but promote genomic stability. They ensure the DNA repair pathways. So they ensure that the DNA repair pathways are working all fine, so that the cell won't lose their control over the cell division, and now there will be no formation of tumor. So the caretaker tumor suppressor genes, they ensure the proper working of DNA repair pathways. Okay. So the genes which are involved in DNA repair pathways, they are known as caretaker genes, while the genes which are directly involved in tumor suppression by activating the apoptotic pathways, those are known as gatekeeper genes. So the tumor suppressor genes, they inhibit the cell cycle progression. Okay, so now if there is no cell cycle progression, there will be no M phase and there will be no cell division. So this is how they inhibit cell proliferation. So, and they alongside that, they also control the cell cycle checkpoint proteins, the CDK proteins and the cyclin proteins. So they encode the cell cycle checkpoint control proteins. They promote apoptosis, and they also they also have a role in DNA repair pathway by by gatekeeping the enzymes which are participating in DNA repair. So overall, in short, tumor suppressor genes, they are involved in DNA damage repair, cell cycle checkpoint activation, and cell death. Okay, so they're generally the recessive mutations are the cause. They generally the recessive mutations are seen in the tumor suppressor genes. So you need both of the copies of genes, tumor suppressor genes, to be inactivated for the causation of cancer. So let's summarize the whatever we have learned today. So there are the genes. So we learned about the two hallmarks of cancer. First, the sustaining proliferative signal, and second, how the tumor cells they are able to evade growth suppression. So in that, we learned about certain cancer critical genes, the genes which are very important for progression of cancer. Those genes are known as cancer critical genes. Okay, cancer critical genes or cancer related genes. So there are two types of cancer critical genes that we learned today. The first ones are proto-oncogenes, and the other ones are tumor suppressor genes. Both of them are antagonistic to each other. They have the exact opposite roles. So the proto-oncogenes, upon gain of function mutation, they gets converted into the oncogenes, which are the drivers of the cancers. Okay, while the tumor suppressor genes, they when upon the loss of function mutation, they result into the cancer. Okay, so this the mutations that which happens in the proto-oncogenes, they are usually dominant mutation. Even if the single allele of proto-oncogene is mutated, that is going to result in the cancer. While the tumor suppressor genes, they usually exhibit recessive mutations. Okay, so you need to mutate both of the copies of the genes. So you need to mutate both of the alleles which have the tumor suppressor genes so that it will lead to the progression of cancer. So the proto-oncogenes are the genes which are going to stimulate the cell growth by producing the growth factors, by producing growth factor receptors, or the transcription factors which are important in cell division and cell differentiation, or the signal transducers. Okay, while on the contrary, the tumor suppressor genes, they are involved in DNA damage repair pathways. Okay, by ensuring that DNA damage repair pathways work fine, they ensure the genomic stability. They also produce the cell cycle checkpoint control proteins. So they are involved in cell cycle checkpoint activation, and they are also involved in cell death. Okay, so they also keep control over the apoptotic pathway. So I hope everything is clear in this lecture itself. So that's it for today. In next lecture we will learn about the examples of those oncogenes and tumor suppressor genes, and we will learn about how they are able to result into the cancer. So we will learn about the retinoblastoma. We will also learn about the BCR ABL translocation, which results into the CML. So we will learn about those. We will also learn about the certain signaling pathways which are present in normal cell, but due to certain mutations, how that results into cancer. So we will learn about RAS signaling pathway. So in short, we will be learning about the mechanisms of cancer in next lecture. Till then, see you. Bye bye.